interruptions of normal
Live
operation.
streaming is on.
And there is, of course, last but not least, that we took inspiration from pioneering thought-provoking work and ideas that also helped to shape our own projects, and work that set the path to see how we take advantage of ecological concepts and ecological theory for the benefit of environmental biotechnology. So our main research interest centers around the drivers of community assembly. I think we can agree that the microbiome is the engine that powers environmental biotechnology process. But as we know, contrary to the electrical engines, the microbiomes do not assemble for the addition of individual components, but they undergo a process of self-assembly. And we also know that the activated sludge contain an extremely high number of different microbial species that work together to carry out the environmental service, as this has been illustrated quantitatively in this very extensive survey of 1,200 activated sludge samples from practically all over the world. And we also know that bacteria are not only in high number, but they also span a very wide range of phylogenetic diversity, as it has also been shown by several studies, including this very extensive survey of more than 100 activated sludge microbiomes. So the main question, the main objective in our lab is to try to integrate the environmental and process conditions and also biotic interactions to understand how is the process of a microbiome assembly in environmental biotechnology process, and how these different community compositions influence process function and stability. So I'm going to discuss briefly 3 different projects from our laboratory in which we address this question of community assembly, and specifically we ask how microbial population responded to the biotic factors such as difference in substrate composition and disturbances, and also to biotic interactions with the phages that are predators that attack bacteria. And we performed these studies in full-scale systems, which, although they have certain limitations because there are variables that cannot be manipulated, they provide realistic conditions that are particularly relevant for processes in which scale has a dramatic effect. And I will start by discussing the results of a study in which we evaluated bacterial communities in full-scale wastewater treatment plants that were sampled at different times, that included industries from different activities, and we also included domestic wastewater treatment plants. And we also we generated our own data sets, and we compare this data with other data obtained from wastewater treatment plants, industrial and municipal, obtained by other laboratories in other parts of the world. And we were able to associate the taxonomic and also functional differences between treatment plants to the differences in substrate composition. So that means that each industrial wastewater treatment plant exhibited a unique bacterial composition that clustered according to wastewater type. And and it was clearly distinct in the case of municipal domestic wastewater treatment that formed a specific cluster. And what was remarkable about these results is that they were robust against with respect to plant configuration, operational conditions, and even to laboratory protocols. That is, different DNA extraction methods, different sequencing platforms. Then we focused on a municipal sewage treatment plant and performed the longitudinal study. This is one of the biggest plants in Argentina. It serves a little above 500,000 people. It treats only domestic wastewater. There is no industrial wastewater coming into this plant. And the temperature range between winter and summer is not too large. The sampling included was performed for about 3 years and included a time of a plant upgrade that involved civil engineering works that defined a period marked by events of operational anomalies, which included short-term interruption of feed supply, fluctuation in solid concentration, and some relatively long periods of very low dissolved oxygen concentration. The arrows point to these individual events, and all the period is marked on this with yellow. The sampling also included a period of a highly stable operation that is marked here in green. So each of these periods span a time of about 9 months. And this study comprised the analysis of 60 metagenomes that were acquired across the time series of about 3 years, and the reconstruction of more than 250 genomes from those metagenomes. What we saw is that the distribution of functions and genomes and taxa across the time series define at least two very distinct group of samples that are also here shown in green and yellow that had a very close correspondence to the two periods of disturbance and stability. We can see the dynamics of these bacterial populations in this somewhat psychedelic plot in which each color represents a different genome. It is probably hard to see here clear differences, but if we translate this plot into a plot showing the number of copies of the ribosomal RNA operon, what we see is something like this. Now we can see that there is a clear change in the relative proportion of bacteria according to the rRNA operon copy number. And why is this important? It's important because the number of ribosomal RNA operons in bacterial genomes is what predicts two very important components of growth, of reproduction. That is the growth rate and the growth efficiency. How fast they can replicate and with what efficiency they can use the available resources. And these two parameters are favored under different and contrasting regimes of resource availability. So it is not surprising, it is completely expected that under stable conditions, the community composition will be dominated by bacteria having only one copy of the ribosomal RNA operon. That is because those bacteria are adapted to live under very low resource conditions, which is what we have in a stable medium in which BOD is very low. That is, the carbon concentration is limiting. And that changes under this, in this case, we saw a change under disturbed conditions, and a very clear increase in bacteria having 2 or 3 copies of the rRNA operon. These differences are plotted in this plot, showing that this there is the numbers of copy of the rRNA operon are statistically significant. But then we wonder how if the wastewater treatment plant could adapt to these disturbed conditions and continue to service with a relatively good efficiency. Why is that? How can we explain the dominance of bacteria with a higher number of rRNA operon copy numbers when the resource availability has not changed? And then we measure we estimated the replication index, that is a proxy for the growth rate, maximum growth rate of bacteria in across the time series from the metagenomic samples. And what we observed is that there was we did not observe differences in the estimated replication indexes across the time series. So we speculate that, and we know from this paper by Levine et al. That the decrease in the number of copies of this operon not only influences growth rate, but also influences the length of the lag phase. So given that we did not observe differences in the estimated replication index, we speculate that shorter lag phase provide bacteria with a competitive advantage to rapidly adjust their growth under unsteady operating conditions. So I will spend the rest of my talk switching to what are the most abundant entities on Earth, that are the prokaryotic viruses or phages, that of course are also present in very high numbers in activated sludge. This was determined more than 40 years ago. In 1980, there were 2 papers were published that they showed that the numbers of phage, the ratio between phage and bacteria was about 10. So that means there are approximately 10 viruses for each bacterial cell. And then we know lots of things about phages. We know that they kill bacteria. They know that they may control gene expression, bacterial gene expression. They can even transfer genetic material between different cells. And by killing bacteria, they are also changing the relative abundance of different cells in a community, so they affect microbial diversity. So while we know lots of things about genetic things about phages, we actually don't know exactly what are their role in activated sludge. There was a paper published just a few years ago, a study by the lab of Tom Curtis, that showed that phage abundance was correlated with the total bacterial abundance, with the ammonium oxidizing abundance, and also, very importantly, with as shown here in this correlation table, with effluent ammonium. So these results suggested that the phages actually do have an important role in controlling bacterial abundance, maybe bacterial community structure, but also a functional stability. The thing is, it is difficult to study. It's not that we don't study phages because we don't think they are important, but the thing is we didn't have the tools to make high resolution studies of phage. And one specific problem is or challenge is to identify pairs of a host phage. That means to assign which bacteria is infected by which phage. And possibly one of the most suitable tools for the identification of phage host pairs is the use of the sequences of CRISPR arrays. As you know, CRISPR is an adaptive prokaryotic immune system that provides acquired immunity against successive invasions from any DNA, could be phages or could be plasmid to a cell. And this figure here shows the mechanism by which bacteria acquires the immunity. There is what is important here for us is that in the first step, the acquisition step, the Cas enzymes in the CRISPR array or CRISPR locus copy a sequence from the phage genome, that would be the protospacer, and it incorporates that sequence into the CRISPR array that would be a spacer that is separated by other sequences that are the repeats. And so if we know, if we can identify a sequence in the CRISPR array corresponding to a spacer that is 100% identical to a sequence in a sequence of a phage, we know that that phage has seen that bacteria. So they have seen each other. This phage is the one that can infect that bacteria. And so in that way, we identify this corresponding pair. Using that tool, we were able to reconstruct the dynamics of several phage host pairs. And this is exemplified here with Gordonia, a common bacteria present in activated sludge, frequently associated with the formation of foam, that has this profile across the time series. And you see the green one that corresponds to the phage that follows that dynamics. And there is a second phage that is also infecting the same bacteria that has only a single peak here and one much smaller later. But we can not only reconstruct the dynamics, but there is more information that can be extracted from this, and that uses the fact that the incorporation of spacers that not occur at random within the CRISPR locus, but new spacers are incorporated always unidirectionally adjacent to the leader end of the CRISPR locus. And that provides a historical record of interactions between bacteria and phages. I will explain this in the following with the following cartoons. We have a cell that duplicates. Of course, the daughter cell will have an identical genome, including, of course, the CRISPR locus, which is the region that is shown here. So we have here the Cas enzymes, the leader sequence, and then the repeat spacer. When after duplication, these two cells will distribute in the aeration basin, and they can be exposed to different phage attacks. And then in each case, there may be an incorporation of a different spacer. So these two cells that will differ in the CRISPR locus in this position. And this may happen all over again with the successive infections, and giving origin to a very high, making a diverse population of CRISPR arrays that come from a single cell. And then to make sense of these informations, what we can do is we can condense the information by considering the spacers as nodes that will allow us to build the network. We don't need the Cas enzymes. We then collapse the nodes that are identical, that are the same. And this is how we build a network that in real life looks like this. So this is how it looks when applied to the sequences of Gordonia in the longitudinal study and with the dynamics that I just shown. We were able to reconstruct these CRISPR populations of Gordonia present across the whole time series. And what we see here is an almost clonal conservation of the trailer end, what is called the trailer end of the CRISPR locus. And the gray color indicates that the sequences of these spacers, each of these circles represent a spacer and the nodes of the network. And these spacers, they do not match any circulating phage. And in opposition, the incorporation of new spacers gave rise to multiple coexisting populations. So each of these branches is a different population. So there are main populations that are more abundant. That's why the circle is a little bit bigger. And there is the incorporation of spacers follow these different paths. And so most of what is remarkable here is that most of the new spacers match to sequence that of a phage that is circulated, that is also present, is coexisting with the bacteria. And so we can focus on these main branches, the one that has larger circles, and plot the abundance of each one of these variants along the time scale. And what we see is that the main variant is this one represented here in red. And when we see here in this network, what we see is that the variant lacks the CRISPR immunity against the phage. This variant does not have any spacer that matches the phage genome, meaning that it doesn't have a CRISPR immunity. You see here is represented the red population against the total population, and you see that it's almost 50% along the whole time series. So that means that the long-term phage host coexistence is established through the diversification of the CRISPR locus, but also by the dominance of a population that is susceptible to phage infection, and that allows the coexistence to take place. So, what do the results that I have shown have in common, and what do they tell us about the activated microbiome assembly? What we see is that there is a functional microbiota with the capacity to adapt to a wide range of environmental conditions. They adapt to changes in substrates. They have a flexible response to disturbance, and they can coexist with the predators. So these are three examples of ecological plasticity. Not only the fact that they can be generalist, but they are also have a flexible response to environmental conditions. So there is a wide repository of genes that enables this plasticity of the wastewater treatment plant microbiome. Then, of course, we still need to follow up on these concepts and maybe design experiments that can test these ideas quantitatively. But I think that there is also a take-home message for practitioners, and that is that we need to see fluctuations as an expression of the ability of the microbiome to adapt and to adapt in order to ensure optimal function under these conditions that are the dynamics. And this concept may help to the more conservative thinking to move away from this famous expression, because we do have something to fix. We need to address the new challenges of making the systems more sustainable, for example, by incorporating food waste to the digesters that will have will affect the upstream conditions of the system, as it is already done in many parts of the world, and other changes that may alter the standard operation of the existing system. So there is a possibility to move forward and taking into account this ability that has the microbiome to for the dynamic adaptation. And so with this, I would like to thank the talented people in the lab that did the work I told, as well as acknowledge our collaboration with the water company, ISA, and also to thank the national institutions that funded these projects, and I thank you for your attention.
Thank you, Leonardo. I hope you're online. You can switch on the camera, and also participants can switch on the camera if they like, because then we can see your faces.
Yes.
Any questions for Leonardo? If you're online, just open your microphone and start speaking if you cannot, so don't be shy. Yeah. Leonardo is online, or?
Yeah, can you see me? Can you hear me?
Yes, no, we can hear you, yes. We were just wondering.
Can you see me too?
Now we see you, I guess.
Okay.
No one else. You guys can hear me?
Yes.
All right. Thanks, Leonardo, for a great presentation. I was wondering, when you showed that example of the full-scale wastewater treatment plant and the perturbations, right, that happened there, you had the different RNA operon copy numbers, which came out, differently during stable conditions and then disturbance conditions, right? So, what do you think, based on your experience, about using disturbance conditions on purpose to kind of make the community get used to perturbations, right? If you know some perturbations will occur, for example, for industrial wastewater treatment plants, right? Maybe you can use perturbations on purpose so they respond faster. Do you have any thoughts on that?
Yeah, I think that's a great idea. I think that has been put forward by the theory of microbial, the MRR, I mean, that concept is in line with I had just shown that you can take advantage of the ability of the microbiome to adapt to perturbations. So it will probably increase diversity and have make it the possibility of respond faster if the system is already adapted to this constant of previous disturbance. Yeah, that's definitely is I think it's an idea to that can be used for better adaptation. Yeah.
Thanks.
Thank you. Other people want to address one of the points, or? Okay, that's really nice now.
Hi, Leonardo. Thanks for a great talk. I really enjoyed it. The RNA operand stuff is really interesting, but is it cause or effect? So is the disturbance causing the changing copy number, or is the changing copy number a response to the disturbance?
I think it is a response to the disturbance, because I didn't I didn't go into details of what there were external disturbances when there were practical things related to the fact that the plant was undergoing an upgrade, and so that was a response. It's not that the system started to operated at suboptimal conditions because the community has changed, but it was the community that changed, and still, even though the output was not optimal, it was relatively acceptable for it. But I have to maybe I have to warn that this system only treats is for carbon removal. So because we did see that the nitrogen oxidation was affected by these conditions.
Thank you.
Thank you, Tom. And Leonardo, again, thank you for your nice contribution and really interesting observations. Please stay here for the stay online for the roundtable at the end. Then there's more time for discussing. I now will give the rest of the introductions to my co-chair, Zedeng.
Thank you. Let's welcome the next speaker, Ezequiel Santillan from Nanyang Technological University. He will give us a talk with the title Bacteria Community Assembly Predictably Precedes Changes in Diversity Under a Gradient of Disturbance Frequencies in Wastewater Treatment Reactors.
All right. I want to thank all of you who are here for this session, and also to the organizers for the opportunity to present at what is my favorite conference. So the main message of this talk is actually spoiled right here in the title. But let's see anyways, what does this mean and why we think this is important. So most of us in this conference believe that microbial ecology and water engineering go hand by hand. But for some of you, maybe your heart beats faster when you think of the practical side of engineering, while others might be more attracted to the appeal of microbial ecology. And I wish I could see some of you are nodding right now. Well, the topic of this session should help bring both parties a little bit closer, because our water engineering systems depend on complex microbial communities of living organisms that interact with themselves and their environment. And in that regard, ecological theory can help us draw generalized conclusions from specific observations of these organisms in their environment by allowing us to classify, interpret, and predict the world around us, which is desired for engineering. And I'm saying this being an engineer myself. So the focus of this work is on disturbance, which is important in ecology because it structures ecosystems affecting their functions. In water systems, this can be the result of a leak or the addition of pesticides, antibiotics, or chemical compounds. And a disturbance is defined as any event that kills, harms, or inhibits members of a community, but at the same time creating opportunities for other organisms to grow and reproduce. Succession, which is the sequential replacement of a species, like in this image after a fire, often occurs after disturbance. And moreover, disturbance can lead to alternative stable states that may or may not be reversible. So at Celsi, we worked with two types of environmentally relevant disturbances using activated sludge reactors. One is trichloroaniline, which is a xenobiotic compound, and the other one is organic loading shocks in the form of double chemical oxygen demand addition. And we did experiments at the microcosm scale with replicated sequencing batch reactors subjected to different frequencies of disturbance, either pulses or continuous, for periods to 5 to 6 weeks. And then we further experimented at a 2 orders of magnitude bigger scale of volume and longer periods of operation of around 5 to 6 months. The reason why we did this is because disturbance is multidimensional. So understanding its effects is actually tricky, as it can vary in type, intensity, frequency, and its effects can be scale dependent. So all right, for now, let's focus on only these two studies. What do you think happens when activated sludge reactors are subjected to different frequencies of disturbance? Ecology, outcomes is the intermediate disturbance hypothesis, or IDH for short. This hypothesis predicts a peak of diversity at the level of disturbance due to a competition colonization trade-off that organisms face. However, the IDH only explains diversity, which is just a structured portion of the community. This leaves gaps related to what assembly mechanisms and the ultimate impact, if there's any, on ecosystem function. So we hypothesized about the role of stochastic and deterministic assembly mechanisms as shapers of community structure based on observations using different frequencies of trichloranilene addition as disturbances. We proposed that when intermediate disturbance regimes result in less predictable environments, is the stochastic equalization of competitive advantages what leads to the observed higher alpha diversity. On the contrary, on extreme ends of the range, where conditions are recurrent and therefore more predictable, this would select for adapted organisms whose dominance would result in a lower alpha diversity. But again, disturbance is multidimensional, and that was only one study. So we tested this new hypothesis with a different type of disturbance, double organic loading, 5 replicates instead of 3 for more statistical power, and for a total of 42 days. The experimental design employed microbial sludge from a full-scale facility, which was used to inoculate 30 microcosm reactors of 25 milliliters of working volume. So these reactors were operated in one-day batch cycles where complex synthetic wastewater was fed daily to mimic the complexity of real wastewater, while avoiding the normal composition fluctuations and immigration of microorganisms, so as to better control the factors influencing our observations. So results from 16S ribosomal RNA amplicon sequencing show that after an initial decrease in diversity from that of the full-scale plant inoculum, we found again a parabolic pattern of alpha diversity, this time from day 21 onwards, which validated again the intermediate disturbance hypothesis. Yet further, higher alpha diversity at intermediate levels of disturbance was observed for both taxonomic and phylogenetic indices, both unweight and abundance weight. And this is relevant because the IDH originally was only defined for richness. Moreover, when we quantified assembly mechanisms using the NIREX taxon index null modeling approach, we found a significantly higher contribution of assembly mechanisms at intermediate levels of disturbance, which supported the intermediate stochasticity hypothesis. There was also a significant positive correlation between the relative strength of stochasticity and all the alpha diversity metrics that I showed you in the previous slide. But more interesting was that the parabolic pattern in stochasticity actually preceded that of alpha diversity in time. So the temporal dynamics of beta diversity showed that communities diverged over time across the range of disturbance frequency. And this resulted in the formation of 3 significantly different clusters at undisturbed, press-disturbed, and intermediately disturbed levels. Misclassification errors of canonical analysis of principal coordinates showed that replicate reactors were less similar to each other at intermediate levels of disturbance, while replicates at low and high disturbance frequencies were more similar. That's why the misclassification error is 0. Likewise, community assembly assessed via the beta-NIREX taxon index showed a higher relative contribution of stochasticity at intermediate levels of disturbance, with beta NTI values closer to 0, indicating that phylogenetic turnover across within treatment replicates was closer in this case to the null expectation. So general functions of carbon removal and sludge settleability actually improved over time of the study. Similarly, the overall strength of deterministic processes increased with time. Hence, correlations between these functions at NTI were negative and significant, which indicated an improved performance of these general functions under stronger deterministic assembly mechanisms. Further, carbon removal was better when alpha diversity was lower. So a more diverse community did not necessarily translate into a better ecosystem function in this case. On the other hand, nitrogen removal via nitrification was positively correlated with stochasticity, being higher at intermediate disturbance frequencies during the initial stages of the study, where stochastic processes were more prevalent. Such correlation disappeared, however, at a later stage of the study. So we believe all these findings are very encouraging because they show for microbial systems that changes in community assembly actually precede changes in community structure. And on top of that, stochastic assembly operating at intermediate levels of disturbance could be the reason why higher diversity does not necessarily mean a better function. So these ideas could be then applied to, for example, identify cases when disturbance-induced stochastic assembly promotes alternative stable state of community structure that either compromise or enhance ecosystem function. So then we can design mitigation or intensification strategies accordingly. It could be used to develop functionally resilient communities that do not occur naturally via the stochastic mechanisms that are triggered at intermediate frequencies of disturbance. And they also hold potential for a general understanding of disturbance-induced changes in community structure and function by integrating the influence of the underlying assembly mechanisms. But since disturbance is multidimensional, for this we call for more research on different microbial systems, types of disturbances, and scales. So this preprint over here describes in detail the work that I have just shown, but I would also like to invite you to check out some of the other aspects of ecological theory for disturbance that we have explored at Celsi using activated sludge reactors. And with that, I would like to acknowledge my PI and former PhD advisor, Professor Stefan Wertz, as well as many others who have also provided support for the work I referred to during this talk. And thank you all for your time, and I'd be happy to take any questions.
Thank you very much for your presentation. I think we have time for 2 questions. We already have one question from Joseph Weaver. So would you like to ask yourself?
Are you online, Joe?
Okay.
Sorry about that. I had a hardware mute again. Great presentation. I really liked the energy. I know I'm asking you to sort of suppose here, but I'm really curious. How do you think these communities would react to repeated instabilities? So after the 3rd or 4th time a community saw a COD shock load, do you think it would be thrown as far into a stochastic assembly process, or do you think it might be a little bit, for lack of a better word, calmer when that happens?
Thanks for the question. So based on these 2 studies, right, the one I described more in detail now and the one we previously did with trichloroanilin, it seems that stochastic processes are triggered at intermediate frequencies of pulses, right? So the closer you are, I mean, in the range that we use in these studies, which is very extreme because it goes from an undisturbed condition to a press disturbed condition, right? In those cases, the communities, in comparison with the ones at intermediate levels, behave more deterministically. And then this is indicators of community assembly, also beta diversity in terms of structure, and then in function in some cases. So that answers your question. And then on top of that, we've seen this effect of higher alpha diversity at the intermediate levels. So it seems that within a range, there's a certain degree where pulses will elicit stochasticity, and perhaps that could be used for some beneficial purpose. If we could trigger, alternative stable states that could be beneficial somehow.
That's what I was thinking. Like if you could just keep on shocking it until it behaved better.
Exactly, and then that exactly. So in theory, that could lead the communities to an uncertain, and I mean, following the stochasticity, right, which means that there is some uncertainty, sorry, that could be described with a probability distribution. This could lead to an alternative stable state that would only be triggered by disturbance, and that could be something beneficial, or not.
Any other questions from the audience on site? Yes, Timmy? Maybe switch to this one? Yeah.
Eric, can you increase the volume of the stand microphone?
Hi. Yeah. Hello. Hi, Ezequiel.
Hi.
Very nice presentation, and I'm wondering how to actually use this knowledge that you presented in the practice, because I feel that we work, at least from my perspective, I see a lot of work is being done in enrichments in the lab. So those are pretty stable conditions in which we see, and then we study the organisms, and we get certain principles from these enrichments in the lab. But actually, I think in practice, there's a lot of disturbances going on, right? So do you think, based on your study and the stochasticity that you showed, the knowledge we get from enrichments in the lab would actually misguide us because we study organisms in really stable conditions? And would that maybe lead to, would you suggest that it's better to study organisms in the plants directly, or to make enrichments with these kind of disturbances in the enrichment itself?
Thanks. I think it's a great question, and also a question that I do to myself. When we evaluate these theories, right? If we want to evaluate them right, we try to control as many factors as we can, right? And that's something we could do, especially to detect assembly mechanisms. We had a discussion yesterday when if you want to capture variability, it's better to have replicated systems, right? And then you can do that more easily in a microcosm setting than you could do with full-scale systems, right? So doing this, we increase, let's say, the internal validity of our observations, right? I mean, the highest version of that would be to just do mathematical models. And then what I think we should do is to have, for example, conversations like this one. I mean, this session, there are several speakers that will present and will present results in full-scale systems. There's a little bit of modeling. My system is microcosm, right? And then I think these ideas could help us understand better what we see at disturbance conditions in full-scale systems. And then you could be more sure of the observations we saw in the smaller systems because things were more controlled, and maybe that will seed some ideas that when you go full scale and then you see some disturbances, okay, yeah, actually there's evidence that alternative stable states of community, structure or certain functions have been shown in the lab. Maybe this is what I'm seeing here in full scale system, right? But of course you have many more variables. So that's something that I speculate could be possible. So I would say we should have a conversation between people that are, and in ecology also happens, people that field ecology versus people working, with the experimental setups. And then we try to find or get together somewhere in the middle, because none of the cases will fully explain what's really happening.
Yeah. Great, thank you.
Thanks for the question.
Thanks for the discussion. I think we can move on to the next speaker and keep this discussion in the breakout session. So we have the next speaker, Giulia Dottorini from Aalborg University. She will introduce us the temporal and spatial impact of mass immigration for the assembly of microbial communities in wastewater treatment plants across Denmark.
Hello, everyone. My name is Giulia Dottorini. I'm from Center for Microbial Communities in Aalborg University. Today, I'm going to show you some results from our study titled Temporal and Spatial Impact of Mass Immigration for the Assembly of Microbial Communities in Wastewater Treatment Plants across Denmark. The activated sludge process in a wastewater treatment plant is carried out by complex microbial communities, mainly composed by bacteria. They sustain the whole process of wastewater treatment and are known to be shaped by both process design, but also operational parameters such as sludge retention time, organic loading, oxygen concentration, and others. However, wastewater treatment plants are open ecosystems, which are designed to continuously receive not only organic matter to treat, but also bacteria. So the passive movement of individuals from an upstream source, such as influent wastewater, to a downstream ecosystem, such as activated sludge, is referred to as immigration. The role of immigration is highly debated in literature, and from a practical point of view, from an operational point of view, it is neglected. But it is, however, assumed to be important. So in our group, we have looked a bit closer into immigration, trying to understand how immigrating bacteria affect the assembly, the community assembly and the community structure of Actioides communities. Where by structure, I refer to both presence and abundance of bacteria. The approach that we adopted is based on the collection of both influent and Actioides samples, which have been sampled at different wastewater treatment plants all over Denmark. We have collected different samples with different frequency over longer or shorter periods, giving rise to different space-time series datasets that we use to address different questions about immigration. Once that the samples have been collected, we have applied an amplicon sequencing workflow using Illumina sequencing, and we applied our Midas taxonomy developed in-house to identify and quantify in terms of relative rate abundance the species present in our samples. So first of all, we looked at the community structure in influent and activated sludge. Here we're looking at a principal coordinate analysis where every symbol represents a sample collected either in influent or activated sludge for 10 days. Each color represents a wastewater treatment plant for a total here of 69 plants. As you can see, the influent and activated sludge samples cluster separately, indicating overall different community structure. We can also observe that activated sludge samples cluster more closely to each other over time, indicating a higher similarity over time compared to influent wastewater samples, which are instead more scattered over the blood. We have also performed the analysis of variance of influent and activated sludge samples separately, taking into account different parameters. And we have observed that the community similarity is higher for samples of influent or activated sludge taken from the same plant. And in particular, the similarity is higher for activated sludge samples compared to influent wastewater samples. We've also investigated the possible effect of geography and found that both influent and activated sludge communities are not very much influenced by which region in Denmark the samples were taken from. Since we have observed that the community structure in influent and activated sludge were overall different, we have evaluated the fate of immigrating bacteria in activated sludge by means of mass balance that we have used to calculate the specific network rate of bacterial species. And we have then grouped the species into disappearing, surviving, or growing. If we look at the cumulative rate of abundance of the species in influent, then we can see that the biggest fraction is composed by species that disappear in activated sludge, while the smallest fraction is composed by bacteria able to grow in activated sludge, which include also the process-critical bacteria for the activated sludge, which are then present in very low abundance in the influent. On the contrary, if we look at the fractions in activated sludge, then the biggest fraction of the cumulative rate abundance is composed by growing species, as expected. Since they are the ones that are transported by the influent and then are able to thrive and grow in activated sludge. However, in activated sludge, still more than 10% of the relative rate abundance is composed by disappearing species, which are probably detected in such a high abundance because continuously immigrating in activated sludge through the influent. So finally, if we consider one specific species and its fate, then we have found that, yes, its fate is approximately the same in different wastewater treatment plants that we have considered in the study. In particular, we have taking into account 11 plants with all similar design, so all with phosphorus and nitrogen removal, where the fate of the same species was consistent. Then we looked at how immigration potentially affects the identity, the presence of bacteria in activity sludge. Here we're looking at the top 200 most abundant species in activity sludge of each of these 11 plants that we have included in the analysis. And in red is indicated if that species is found also in the corresponding influent wastewater from the same plant. And as you can see, the most abundant species in activity sludge are also present in the corresponding influent. And we have observed also similar results for unique species in activity sludge of each plant. This overall indicates that immigration creates a sort of microbial fingerprint for the presence of bacteria in activity sludge. We also evaluated the possibility to predict the abundance of species in activity sludge by knowing the abundance of species in the influent. And we have found that yes, this is possible. And we obtained two different predictive patterns using two different statistical modeling. So in the first case, if the prediction, when the abundance of a species in activity sludge was well predicted by the univariate model, we refer to such a prediction as the univocal association, which in the case of Streptococcus parasuis indicates that the abundance in activity sludge of such species is well predicted by its own abundance in the influent. When instead the abundance of a species in activity sludge was well predicted by the partial least squared regression model, then we refer to such a case as interspecies association. An example is for the species Tetrasphera midas S5, where we have interpreted the result in the sense that the abundance of the species in activity sludge is predicted by the abundance of other species in influent. And we don't know exactly why this is so, but some hypotheses are that there might be some co-immigration of species from the influent, from the sewers. Finally, from a different data set, we have observed the abundance of certain species in activity sludge. In particular, here we are looking at 3 filamentous species and how their abundance vary over time in 1 year in one specific plant. As you can see, every species here has its own trends, its own abundance variation over the year. And if we look at how the abundance of the same species varies in influent wastewater, then we have observed that the abundance actually varies almost in the same way in the influent all over the year or across the 1 year of sampling. So this indicates that these are preliminary results, but indicates still that immigration may play a role in the yearly abundance variation of species in activity sludge. Sorry. And in particular, immigration might help us to understand the dynamics of species in activity sludge. So to conclude and to summarize the results, I think we can conclude that immigration in activity sludge is more important than what we previously thought. We have observed in particular that immigration controls both the presence and the abundance of bacteria in activity sludge, therefore being involved in the assembly of activity sludge communities. By investigating the community in the influent wastewater, we have highlighted the presence of a phase of immigrating bacteria, where the biggest fraction of biomass in influent disappears in activity sludge, while the growing bacteria are present in very low abundance in influent. And also we have observed how the fate of a species is consistent in plants with similar design. Finally, we have observed how for the same species, there is a similar variation of abundance over a year in influent and activity sludge. So with this, I would like to conclude and thank you for your attention.
Thank you very much for your presentation. And if you are online, please turn on your camera. Do we have any questions for Julia? Yes, please.
Is this on? Yes. Okay. I have a question. Can you hear me?
Yes. Yes. Hello. Can you hear me?
We hear
Maybe
you.
not.
No, we hear you.
All right. Hello, everyone.
Hi. Hi. Yes, I'm having some difficulties with the microphone. My question is quick. Yeah.
Yes.
All right. I'm hearing my own voice repeated to me. My question is, have you considered other variables, physicochemical variables, in your modeling other than biotic variables?
No, not in the modeling. So we are aware that there might be some other parameters influencing the community, but the modeling takes only into account the variation of abundance. It's a predictive model that takes into account only variation of abundance. So no.
Hi, I also had a question. So you see the reason I'm asking is that there may be other parameters that are way more important.
I'm sorry. I'm sorry. Can you repeat, please?
So there might be other parameters that are more important.
More important than immigration.
I apologize. This is not working well. I should just let somebody else ask.
I'm sorry. Thank you for the previous question.
Okay, Sam.
Yeah. Okay. So hi, thank you for the talk. So about the fact that most of these bacteria that enter the or immigrating towards the wastewater system are actually disappearing. Does it have to do? I mean, one thing could be that they're maybe not process critical, but doesn't this also give a measure for the inherent fitness of a bacteria that's been discarded by another process?
I think that there is also a matter of selection in activity sludge. So, of course, there are parameters that select for certain bacteria, and some of the process critical coming from the influent are in very low abundance, and they're able to thrive in the activity sludge. But also, I think that the presence of big fraction of disappearing, it is an indication still of selection processes, but also indicates that if we're able to consistently see that fraction of bacteria seem to disappear, then it's also an indication that immigration has quite a role.
No, yeah, with that I definitely agree. So thanks for the talk.
Thank you.
I have a question too. Hey, Julia, thanks. Great work. My question relates to the observation that some taxa, the abundances of taxa in the influent mimic those that you find in activated sludge systems, right? Which would suggest that somehow you're having something similar to an activated sludge process happening in the sewer, which is counterintuitive, right? Especially because it's not the same, right? I mean, unless there are reasons to hypothesize that this could actually be happening, right? So then I find that very, very staggering, right? So how could that be explained from
Yeah, thank you for the question. It's a good point, and I think what our idea about it is that there are some mechanisms happening in the sewers which we are not aware of yet, and for some reasons, this mechanism release possibly microorganisms in a certain way in the sewers, the design of the sewers might also impact the variation of the passive transporters of microorganisms, and this way in which the species vary, maybe randomly, in the sewers is then reflected in the activated sludge. So this is our thought about it.
So you guys would speculate that in the sewer you're having similar, nitrogen removal, phosphorus removal processes, creation of filamentous bacteria, and then of course the carbon removal. That would be the easier one, but the others are a bit harder.
Yeah, no. So that's probably not happening the same way as it happens in activated sludge. We're on that.
Okay, let's move on to the next speaker. Feng Ju from Westlake University. He will show us how to untangle the microbiota diversity and assembly patterns in the world's largest water diversion canal.
Hello, everyone. I'm Feng Ju from Westlake University. It's my great pleasure to talk online at this IWA conference. I'm going to talk about microbiota diversity and assembly patterns in a huge water diversion canal. My postdoc fellow, Lu Zhang, she will join us in the Q&A session. As you know, water resources are not evenly distributed on Earth, and the large canals are developed to deal with water shortages by reallocating water resources for human use, such as irrigation and domestic consumption. Globally, there are more than 110 existing or under construction water diversion mega projects. As quality of the transferred water is closely related to human health and ecosystem safety, it's critically important to monitor and control the canal water quality. Microbes are highly responsive to environmental changes. They are good bioindicators for anthropogenic impacts such as pollution. Moreover, knowledge on the microbial communities or microbiota can provide deep understanding on the key drivers of water quality to inform its management. However, we know little about the microbiota dynamics and assembly patterns in water diversion canals, especially over long geographical distances. The South to North Water Diversion Project is the world's largest water diversion project. It was built to aid in water supply in the drought-stricken North China and consists of 3 routes, namely called Eastern, Middle, and Western routes. So the Middle Route project is over 1,400 kilometers long and served almost 80 million people. The main canal is completely artificial system and is an ideal system to model the microbiota underlying water quality and ecosystem service in the canal. So we asked 3 questions. How do microbial, including bacteria and microeukaryotic communities develop along the main canal? How much does ecological stochasticity, such as random dispersal, contributes to the community assembly? And which immigrating populations can better survive and navigate across the canal? Therefore, we connected water from Danjiangkou Reservoir and 18 sites along the canal, and we applied the quantitative microgene analysis to investigate the canal water microbiota and further use non-model and neutral models to explore the community assembly mechanisms. Firstly, we found that microbial biomass and alpha diversity significantly shifted both temporarily with season and especially along the canal. For example, both bacteria and microeukaryotic biomass was the highest in August, whereas the lowest in October. Moreover, beta diversity analysis revealed strong clustering of microbial communities in relation to seasons. During the year, May and August had larger intra-season community variations. Notably, season is a more pronounced driver of the community variation than special effects along the canal. Microeukaryotic communities appear more taxonomically dynamic along the canal water flow than bacteria. For example, we noticed that metazoan, mostly Canonoda, were dramatically enriched upon entering the canal, but they quickly decline in the downstream sites. So next, we try to address the deterministic factors driving the observed community patterns. So microbial community can be deterministically affected by environmental or abiotic factors. In the investigated canal, we identify water temperature, pH, dissolved oxygen, total nitrogen, and fluoride as co-influential factors of microbiota composition. In addition to deterministic factors, the apparent microbial dispersal with the water flow and the major hydraulic manipulations in the canal, such as inverted siphon and tunnels, drive us to question on the role of stochastic process in the community assembly. Normalized stochasticity ratio analysis revealed contrasting trends in the contribution of stochastic process to bacteria and microeukaryotic community assembly. We also applied slow model and found the neutral process could explain different proportion of community patterns depending on seasons. For instance, their contribution to bacterial assembly is much stronger in March, whereas much lower in May. Especially the model can be utilized to link the overall community patterns to divergent behaviors of different populations by differentiating populations that strictly follow or deviate from the model predictions. As you can see, the three population groups have quite different taxonomic composition. So in general, our study enhances understanding regarding the canal water microbiota is more driven by stochastic process or under which conditions deterministic processes play an increasing role. So to withdraw which immigrating populations die off or active grow in the over 1,000 kilometers canal, we propose the local growth factor LGF as a quantitative measure of variation in the absolute microbial abundance in each canal section relative to the canal head. In this way, a LGF value larger than 0 indicates population growth. Overall, the results suggest that the immigrating microeukaryotes are better adapted to the canal water environment than bacteria. In the bottom figure, each dot represents a microbial population and its color indicates absolute abundance. Overall, we found high abundance population tended to show very high or very low LGF values. For example, 2 abundant bacterial species and 2 uncultured microalgae species show high LGF values that suggest their strong enrichment during water diversion. In stark contrast, 2 cyanobacterial lineages and 3 potentially pathogenic Actinobacter species showed extremely low LGF values. That's around -6, suggesting their severe growth suppression and decay in the canal. Most importantly, our results demonstrate the power of local growth factor in identifying key players and predicting microbial fates during long-distance water transfer. In summary, our study uncovered the community dynamics, assembly mechanisms, and population fate in a giant water divergent canal. So we found that bacterial community is primarily driven by combined environmental effects. And stochastic process in microeukaryotic community needs to be considered in water quality monitoring. So our proposal of local growth factor analysis facilitates identification of key players in the canal system. For example, we identify the sharp decrease of potentially harmful cyanobacteria and potential pathogen, as well as the favorable enrichment of nemohabitants as scavengers of algae-derived organic matter. So the middle root main canal of the south-north water divergent projects is an engineering system, while the aquatic system is still at its early forming stage, and more relevant research is needed. Here, I would like to thank my team members, funding agencies, and collaborators. The bottom photo was taken during our first field campaign in October 2018. It was lots of fun. I would like also to quickly advertise my lab. We are interested in environment microbiome research, and our ultimate goals are to engineer microbes and microbiomes for improved sewage resourceization, bioremediation, and human health protection. That's all, and thank you very much. I'd like to take your questions.
Thank you very much, Dr. Zhu. I think we can ask you to turn on your camera, or maybe Dr. Zhang, for questions. Do we have any questions from the audience or from online attendees?
Hello, everyone.
Hello, we can hear you.
Yeah.
Any questions? Okay.
Okay. Can I ask? I'm sorry, but all the
please.
talks are so interesting. So that's why I keep asking questions. I'm biased, of course, but the question I have for Fenju, thanks a lot for the talk as well. The question I had is related to the difference you saw in assembly mechanisms for the eukaryotic fraction versus the bacterial fraction, right?
Mm-hmm.
So the data suggests that the eukaryotic fraction is driven more stochastically compared to the bacterial one, right? So stochasticity is stronger there. And then you said that according to other data, the microeukaryotes were better acclimated to the water canal compared to bacteria, which would suggest the opposite, right? So so then the question that comes to me is like, what are the reasons why you would speculate that, what are the underlying assembly mechanisms responsible for the detected stochasticity in eukaryotes, right? So what could be triggering that stochasticity in the eukaryotes if they are better acclimated than bacteria?
Mm-hmm.
Yeah. Thank you for the question. Yeah, I think I will. Yeah, Lu, you want to?
Yeah. So can you hear me? Okay. Thank you for the question. So you asked about the link, how we think why they according to our local growth factor analysis, so they adapt to the canal environment better. And on the other hand, we found, based on the neutral model analysis, that they are more stochastic. So did I get it right? Yeah, so I think we also find it very interesting, but depending on the local growth factor analysis, I think so they adapt better to the canal environment. And this kind of ease them from the selection pressure locally in the canal. And this means that they are less pressed compared with the bacteria, and this release of the selection pressure make them more stochastic. So that's how we think about this.
All right. Just very quick, which of the two approaches do you think is more informative of what's really going on?
Thank you. So I think they are both, to be honest, I think they are both very informative, because the NST approach, so it's more quantitative. So they really tell you, I think approximately which processes are more dominant, and they give you an absolute value. And on the other hand, this neutral model-based analysis, this modeling approach, so they can really differentiate based on the modeling which fraction of the community are fall within the model prediction, and which other fraction or the populations, they are more they deviate from the model prediction. So then you can So go down to these populations, and then you can further understand. So link this model prediction with their properties. So for example, looking at their taxonomy, or looking at their maybe their growth, or looking at their so maybe the ribosomal RNA copies, so something like this.
Or growth factor analysis,
Yeah.
as we proposed.
We did, yes. Yeah, thank you.
Thank you.
Thank you very much for presenting this unique study. I think now we can move on to the next speaker, Jia Mingsheng from Hent University. He will elucidate the competition between heterotrophic denitrification and DNRA using the resource ratio theory.
Eric, there is no sound from the console. Don't see the video either. Is a total blackout. I will go to.
We're having some sound problems. We will be right back.
Hi everyone, my name is Mingsheng. I'm currently a postdoc working at CMAT Ghent University. I'm very glad to share with you my PhD research on the competition between heterotrophic denitrification and DNRA. As we all know, we are facing a lot of global challenges for sustainable development, and nitrogen management is also one of it. A group of scientists have tried to identify our planetary boundary for human development. And as you can see from this figure, nitrogen discharge has far exceeded our planetary boundaries with high risk. One of the nitrogen compounds of concern is nitrate. Nitrate is directly related to the eutrophication problem of our surface water, and it concerns the quality of our drinking water. It also widely used as a fertilizer for our food production. And the products of nitrogen reduction has great implications for greenhouse gases emission and thus climate change. There are two major biological pathways for nitrogen reduction. The first one is the widely known denitrification. In this process, nitrate is reduced to nitric gas, during which N2O, which is strong greenhouse gas, can be emitted. Since it removes nitrogen, it has been widely used in wastewater treatment. The other often overlooked process is DNRA. In this process, nitrate is reduced to ammonium. Therefore, nitrogen returns in the ecosystem. This is often wanted for our natural ecosystems, especially for agriculture, for better fertilizer utilization. In the case of heterotrophic denitrification and DNRA, clearly these two processes will compete for both nitrate and organic carbon, or COD, as electron donors. This competition has great implications for the fate of nitrogen and also greenhouse gases emission. So it is important to understand what rules the competition. We first looked into the literature, but the findings are rather confusing. So in general, COD to N ratio is considered as the key factor, and high COD to N ratio generally favor DNRA. And also other researchers found that high COD to N ratio does not necessarily lead to DNRA. And more recently, a study shown that resource concentration rather than COD to N ratio may dictate the competition. So from these seemingly conflicting results, we want to know if we can predict their competition outcomes under different conditions. And we did a modeling study starting from the simplest system. Imagine a chemostat which contains both denitrifying and DNRA bacteria. It is constantly fed with different levels of nitrate and COD. And to predict the competition outcome, in this case, we need to consider both the microbial factors, such as kinetics and genetics, and also environmental factors, including the infant resource concentrations and dilution rate. And we used the resource ratio theory, which was proposed by Takehome for this purpose. In this resource ratio theory, it describes the interaction of competing species through their use and effect on shared resources. In this case, COD and nitrate. It predicts that the species that can survive at the lowest level of a limiting resource will be the best competitor for that resource. Then the question becomes how to define this ability. When it comes to bacterial growth, most often we use the monokinetics shown here. And the growth rate is a function of the substrate concentration of the limiting substrate. And the Ks is the affinity constant and is widely used for microbial competition predictions. But in a chemostat, when it reaches steady state, the growth rate will equal to the dilution rate. From this equation, we could derive the substrate concentration at the steady state as a function of Ks, mu max, and dilution rate. And this is called subsistence concentration, or JS. Previous studies have shown that the lower the JS, the more competitive the species for that specific resources, which means the one with the lowest JS will always win the competition for that resource. In brief, with the input listed here, this resource ratio theory could generate a very straightforward graph to check the model competition, the model outcomes. Here, the X and Y axis are the resource concentrations of the two shared resources. And there are 6 regions with different competition outcomes. With the fixed dilution rate, we only need to know the infant concentrations to make the prediction. For example, if the infant concentrations are in region 6, then the two competing species will be able to coexist at steady state. So we applied this resource ratio theory and got this figure for denitrification and DNRA. To use any model, we need to first validate it. For the validation, we use the experimental data from 2 experimental studies from TU Delft And there they fight the chemostat with different levels of nitrate and COD. And we plotted their observed competition outcomes in this figure with different colors for different competition outcomes. As you can see here, our predictions matched their observations both qualitatively and also quantitatively for biomass concentration. So now we are confident to use this theory to look at the impact of other factors. Since COD-to-N ratio is widely used, here we plot this figure using COD-to-N ratio as the y-axis. Let's first look at the impact of infant COD-to-N ratio on the competition. As we can see from here, with the increase of infant COD-to-N ratio, the dominating process would shift from denatrification to DNA, which is in line with some experimental studies. What is interesting here is that the boundaries are impacted by the infant nitrate concentrations. Clearly, there's substantial impact at low infant concentrations. Also, we found that the same infant COD-to ratio does not necessarily lead to the same competition outcome, as we can see from these 4 points. With the same infant COD-to ratio, they could have totally different competition outcome. And this also raises the question on how to determine the threshold for resource limitation. We also look at the impact of dilution rate and found that there is a critical dilution rate below which the coexistence is possible. And when the dilution rate is lower than this value, it has great impact at high dilution rate or low infant resource concentrations. If we look carefully, researchers often use different systems and often mix the conclusions. Here we demonstrated that for continuous and batch culture, even with the same initial conditions, being the same concentrations of denatrification and DNA bacteria and the substrate concentrations, they could get totally different competition outcome. This reminds us to be careful when comparing the results from different systems, and also when designing our studies. With this, I would like to conclude my presentation with some take-home messages. First, we should always keep in mind the difference between continuous and batch cultures. And in continuous cultures, the true affinity is not Ks, but Js. And in general, high infant COD-to ratio promotes DNA, but the boundary COD-to ratios are impacted by the stoichiometry, infant concentration, dilution rate, and kinetics. Last but not least, this resource ratio theory can be applied to any competition scenarios where they choose a few species compete for 2 shared resources. And we created simple tools to implement this theory. Finally, I would like to thank for your attention. Thank to my 2 PhD promoters and a few other colleagues for their constructive discussions on this work. If you have any questions, do not hesitate to contact me.
Thank you very much, Ming-sen. I think you are online. Please enable your camera. And Mr. Santillan, do you want to have the first question again?
No. Other people should ask questions.
So we have the question from the audience. Maybe Nina?
Thanks for your presentation, Mingcheng. Is he here? Yes, I guess so.
Yeah.
If I'm not mistaken, there are organisms that can do both DNRA and denitrification. Correct me if I'm wrong. How do you then envision this competition? Because then it's not necessarily a competition; it's more kind of a metabolic switch. Have you?
Sorry, I temporarily turned off my microphone because our neighbors just started working on their renovation. So there's maybe high background noise. Yes, you are right. There's bacteria who can do both denitrification and DNA, and this was not considered in this modeling study. So then the question may becomes under which conditions they switch to which pathways, or from which pathways they could get the most energies from. Or if you put, if we call them generalist, that we can do both pathway together with some specialist denitrifier and DI bacteria who will win the competition. And I think this is also interesting study for both modeling and experimental validations.
Thanks.
If there's no other questions, we move to the last speaker of the session. We have Joseph Beaver from Newcastle University. He will tell us why and how drift matters and when it's not.
All right. I remember to turn on my microphone.
You can share your screen.
Are you all seeing the screen?
Not yet.
How about now? Is it visible?
Now we see your initials.
Okay.
No, there's someone else.
Someone else's initials. RS.
Hang on. From beginning.
We cannot see your screen,
At
Michael.
all?
From techniques, there is. We want to project something from an online participant.
Okay. You can also just play my video. I have one updated slide at the end that I can describe. That works just fine.
We will start launching the pre-recording then.
Thank you.
Thank you.
Sorry, we have no sound again.
Hello. I'd like to lead off with a slide I've used before, because the question it asks is central to most of the research I do. Simply put, the question is a matter of getting from point A to point B. Let's assume for a moment that we have some idea of a target community we'd like to achieve. Now, what we'd like to do is have a guide which tells us how to start an arbitrary community and drive it towards that goal. Ecologically speaking, that guide encompasses 4 forces: selection, dispersal, diversification, and drift. You can probably guess from my talk title which one I'm interested in today. There's a bunch of different definitions of drift, which you can read as well as I can, but I'd like to illustrate it for you. The work I've been doing centers around agent-based models of microbial communities. Specifically, I use the NewFeb simulator developed at Newcastle University. In this, each bug is treated as a discrete entity and interacts with the other bugs in a physically and biologically realistic way, thanks to an underlying molecular dynamics package called LAMPS. This figure shows a simple community of 5 identical heterotrophs. Now, what's important here is that there's random elements to their growth, which we can control with repeatability by specifying a random seed. If you change the seed, you get random numbers that are different, and this results in completely different random communities. So why is that? Within NewFeb, there's 2 random processes at work. The first is that whenever a bug is ready to divide, it has to choose a direction to divide in, and it uses the random numbers to determine which direction that would be. Second, when the bug divides, it allocates its biomass between 40 to 60% between each of the daughter cells. That proportion is again randomly chosen. All these random choices lead to identical bugs making very different decisions. And which of those decision sets becomes a winning strategy varies between seed to seed. You can see up here at the top, it's probably a bad idea to move up and to the left because this red one is already dominating. But the same strategy moving up to the left would be a very good strategy under very slightly different conditions. And this gets even more complicated because in reality, this decision is also affected by your neighbor, and then your neighbor's neighbors, and your neighbor's neighbor's neighbors, and so on. The end result is that some colony is generally going to lose. And which colony that is depends on how you initialize the random number generator. Now stop me if you know this story, but I was actually working on a different problem at the time when I noticed this. And that got me thinking. What I had was a simple 5x1 system, and I wanted something with more symmetry to really explore. So here we have my workhorse for this talk. It's a 3x3 symmetric system where all the bugs are equally spaced and for now have identical biologies. The boundaries of the system are periodic, which is a fancy way of saying they wrap around, which is an even fancier way of saying that's like the tunnels at the edge of the map in Pac-Man. I wrote some tooling to track the relative biomass of each colony over time, and this graph is pretty representative of what I see in the different simulations. There's usually a colony or 2 which really thrives, a couple which muddle along and survive along the expected line if all the biomass was equally distributed between the colonies, and a few which languish, and some which would even go extinct if I let it run long enough. Now, the great thing about simulations is that you can manipulate randomness. This has been a huge difficulty in wet lab work when people are trying to study drift. In a simulator, not only can I make everything exactly identically, biologically the same, and identically spaced, but thanks to how random numbers are generated, I can rerun a simulation with the same random numbers, but slightly altered kinetic parameters. This lets me ask my big question. How much of a fitness advantage do I have to give a doomed bug for it to succeed? Here's how I answered that. For a given seed, I identified the biggest loser, which in this case is colony number 4. It's the orange clump near the middle of the front row. The white square just indicates that didn't survive. As you might have guessed from this graph, I then reran the simulation using the same seed, but incrementally changed the kinetic parameters, its maximum specific growth rate or its affinity constant. Here's examples of what happens when I give it a very large boost to its mu max and a slight boost to its affinity constant. The orange clump, formerly the biggest loser, is now thriving, and I mark that down as a black square on my map. If I repeat that 121 times to fill out the grid, you get a map of the conditions under which the doomed bug will beat its fate. Now this is suggestive, but it's really just for one seed. So I repeated that for 120 different seeds. By the way, that means a bit over 14,500 simulations, and I got a bunch of maps. When I combine those maps and average them together, I get what I'm calling a fitness probability map. On the x-axis is the change in mu max, on the y-axis is the change in infinity constant. The color represents the probability that the loser bug will now change its state to a thriving bug. And I've highlighted the baseline case with no change to mu max or ks as a point of reference. Now, a few things pop out to me here. First, if you want a decent chance at thriving, you need to adjust kinetics by a non-trivial amount. Without modifying your ks, your mu max should be increased by about 20% to have a really decent chance of saying, yeah, this bug is going to turn out to thrive instead of be doomed. Same thing for changing the ks without changing the mu. Now, what this essentially means is there's a lot of room in the parameter space for similar heterotrophs to be randomly chosen simply due to drift. Another thing which pops out is that this is essentially a line with a very sharp gradient. Now, while you can analyze this formally, and I plan to do so as a 2D probability density function, when I'm just thinking about what I want to look at next, I like to think of this as an escarpment. Down here is the plane, which is the red area where you're just going to be doomed to die. There's got to be a steep cliff, and then you've got the plateau of thriving. What I'd like to know is how does that cliffside change as I change various things, like when I increase competition by decreasing the space between bugs, or if I lower the ambient substrate concentration. Does that cliff just shift, or does it become a more gentle incline? Does it remain a likely function of mu max over ks, or does it start to curve or even warp around? Based on my initial work just exploring that 5x1 bug system, I can tell you it looks like that cutting the spacing in half at least shifts the boundary down and to the right. So there's even more chance for random drift to control things versus selection via kinetics. At the time I'm recording this, I'm waiting for my larger simulation, which is this 3x3 grid with the space decrease to finish. I expect that simulation to be done by the time of the live talks, so be sure to check in. Now, I love the pointy head microbial biology stuff, but it's a fair question to ask, how is this relevant to an actual physical wastewater treatment plant? In other work I've done, there was a huge, seemingly random turnover in taxa at an aerobic treatment basin. We're talking log 2-fold changes of 5 to 10 for notorious heterotroph generalists like pseudomonads. Yet, if instead of looking at the taxa, I classified things by their function using the MIDAS database and looked at the relative abundance of heterotrophs in the receiver, it was fairly stable, especially compared to the huge swings in taxa turnover. Now, there's a lot of reasons this could be, but one potential factor is that many of those heterotrophs are probably pretty darn similar to each other, and which one is dominant is just the result of winning the spatial lottery I've been telling you about. I'd like to quickly acknowledge my host institution, Newcastle University, and specifically the Rocket HPC cluster. I mentioned that this one run was 14,500 simulations. If I were just doing this on my laptop, then I'd be presenting this talk around May 2023, and that's without much exaggeration. Of course, I'd like to thank the NSF for funding me to do all this work, and I'd especially like to thank Tom Curtis, who's been not only an eager sounding board for all these ideas, but also has gone above and beyond in getting me settled in a new country during the middle of a pandemic. I'd also like to thank Bowen and Dennis. They're the main developers on NewFeb, and they really helped me understand the code base right away and got me productive and hit the ground running as soon as I got here. In closing, I normally ask for any questions, but if you're not watching this live, please feel free to contact me via email or Twitter. I really, really enjoy talking about this stuff.
Okay. So, as Joe said, he really likes to love talking about it. So if anyone wants to give him an incentive.
I love talking about it.
A comment.
And on the screen, I hope I'm showing now is the updated results from the 3x3 grid with bugs spaced closer together. And you see it really did spread out the region, so there's even more room with greater competition. And I'm interpreting that as any mistake you make is even more costly when the competition gets greater due to decreased spacing between neighbors.
Yep. Yeah. Interesting. Any questions in the direction of Joe's presentation specific? And if you're online, just open your mic and raise the question. That's it's not. Otherwise.
Oh.
Yep. There was someone or no.
Hello. Hello.
I
Yes.
have a question. Sorry.
Introduce yourself and then ask the question.
Okay, I'm Claudia Echeverre.
Claudia.
You saw me yesterday from Uruguay in South America. And very interesting your talk. I wonder if this kind of models, it works for biofilms attached to a surface, or it could be also work for other kind of biomass structure as floccules or granules. Okay.
Yeah, that's a good question. Obviously, the biofilm as I modeled it is on a flat surface. I actually found NewFeb by looking at how people model granules and flocks. And the general conceit is that a flock or a granule is just a circle, and you can unwind that to make a flat surface. And as long as you're careful about how you ask the question and your diffusion boundaries and that sort of thing, you can definitely apply it to flocks and granules.
Okay.
But may I add here that essentially a flock is a unit which breaks up and reaggregates all the time, whereas a biofilm and a granule have a more or less constant structure as function of time.
Yes. So not in this, but in NewFeb itself, we have a ability to add fluid flow to it. And one thing I'm interested in is, okay, you've got these randomly selected identical bugs, one of whom is going to really thrive and make this tendril. So what happens when you start treating that as a flock and flow and you get colonizing bits coming off and floating around, and how does that affect things at the macro scale?
Any direct follow-up on this issue?
I got him.
Yep.
Oh, great talk, Joe. I really liked it. And then it really it's a very good complement to the other talks because it's this modeling, right? So I was thinking in this case you evaluated drift, so it's mostly birds and deaths, right? But you could potentially try to evaluate other stochastic processes, like for example priority effects, especially because you have a no, but I think it's very good, right? So the question would be maybe for discussing among everybody, right? Do we have any reasons to believe this wouldn't happen in a full-scale system, right? Do we have any reasons to believe the drift or any of these stochastic processes has absolutely nothing to do with a full-scale system, or actually they can happen and then lead to communities moving up the cliff or down the cliff in terms of a structure, or more importantly in function?
If I can lead off by criticizing my own work here. Obviously, this is a system where every bacteria is the same and it's spaced exactly the same, and there's no other factors happening like immigration from influent or selective settling, or even just priority effects from flocks and granules that are already in the system, right? So what this tool does is it lets you ignore all that. The challenge is then applying those one by one and synthesizing back to the big picture of what's happening in a real world system. And you're absolutely right. I'm actually very excited. Now that I have the system up and running, I can do things like look at priority effects, the size of founder colonies, or even just lag times. The big thing for me is I would really like to get to the point where I understand it mathematically enough that I can make predictions on a stochastically mixed initial setup and then compare that to an actual microcolony grown on a plate.
Maybe a bit related to that, because there's of course quite a bit of presentations around deterministic and stochastic behavior. From an engineering point of view, I would think that deterministic behavior is important for me. Stochastic behavior, well, that's slight variations in some bacteria, but since it's stochastic, they likely do more or less the same. And if they do more or less the same, then I would not be too much bothered by it from a process operating point of view. So is this discussion about deterministic and stochastic behavior of microbial communities mainly a microbial question, or is there also a direct link to process engineering in the form of selection of which you in the end do on metabolic functions? I'm not sure whether anyone likes to comment on this.
Oh, I've got an idea on that. So my former advisor kept on saying, well, if it's stochastic, then do we just throw our hands up? And I have to point out, it's stochastic, it's not random. So you can still influence it. At least the plant I looked at, it looked like startup was very, very much influenced by stochastic processes. And what that means is for your generalist heterotrophs, if you want to get a healthy population, yeah, of course, design the plant according to good engineering practice, but really it's got to be affected by your founding effects. So pay attention to getting a good influence, maybe even evaluate a couple plants in the area, make sure you picked the good one, which, you know, is common sense, but now there's a scientific backing for these rules of thumb that we use.
Someone else want to comment?
Yeah, I would actually like to comment.
Yep.
Okay. Can you hear me?
Yes.
I think also the value of these approaches is that you can explain, you can model and see how the community assembles. I mean, to understand, which is also important to understand the function. I mean, not just what you want to see, but also what is actually there. I find that's there's what the importance of understanding the stochastic processes that are also a component of community assembly.
Yep.
And I would. Who wants to talk?
No,
Yes, I would add that under disturbance, it really matters, right? Unless the plants that are operated by engineers don't face disturbances, but if they do, that could trigger a series of scenarios that hopefully you want to have control on. I mean, unfortunately I never worked on a full-scale plant, but I do know from my friends that are consultants that sometimes the plants fail for no apparent reason, and then they come back to good operation on their own, right? And then that's probably related to what Leonardo said earlier. Okay, if it works, don't mess with it, right? Don't need to fix it, but maybe it's an opportunity for us to understand it better, and then at least get that period where the system is not working well to be shorter.
Do other people have other? Yes, go ahead.
Yeah, well, maybe I would also add, going back to my presentation, that when full systems fail, I mean, we may need to think about the intervention of phages that are in high number, and they may play a role also. There are a few old papers that with anecdotal examples of system failures in which phages may have been involved, and I mean that's some maybe complication that might also be important to add to this the understanding of.
And we give examples how it's, if you have old sludge samples, you might be able to track that down. So you could potentially evaluate it if people would store some samples of the sludge over time. Yeah, David?
I have a question to Julia, to her talk on immigration. And if I understand your findings, this would mean that whatever the operator is doing, you cannot change the community by the operation change of the plant. So it means that everything is governed by the immigration. So could we design an experiment where we feed inert biomass particles that are the same buoyancy as bacteria? And by looking at the recirculation rate of these particles in the plant, we see if we accumulate all these organisms. So in the end, it seems that all these findings on immigration, I think Dominique Frigon, it's a bit the same. How we interpret from an engineering point of view, it's that whatever, if we put aeration or no aeration, if we put anaerobic tank or no anaerobic tank, anoxic tank, no anoxic tank, we just subish what happens in the influent. How do you react to that?
I think it's a bit difficult to say it's totally migration, it's totally selection. It wouldn't be very useful. So I believe it's a combination of both, right? So it is for sure selection. Can you hear me? Yes.
Yes, we hear you, but there was something on the background.
Yeah, sorry, I don't know why. As I said, I think it's a combination, and we have shown that yes, what it is in the plant comes from the influent, and then we start to learn that the abundance of what we see in the plant is related also to immigration. Of course, the growth rate of bacteria is then affected by many other parameters, and of course, the design of the system still affects and is still relevant for the bacteria to be there and do the job, and all the design of POWs and so on still is relevant. But I think we are learning also that maybe there is a chance that if we learn a bit more about immigration, we can potentially address target bacteria before they actually reach the plant and possibly cause problems in the plant. And I'm thinking specifically about microdrix. I think also what we have shown in the recent findings, the last plot I presented, preliminary results, that it seems there is a very good trend of microdrix also in influence. So we have to look closer into that, because immigration is possibly giving us the chance to actually solve problems in advance before they actually happen in activated sludge in the system. And I think there was a second question, which I lost.
Yeah, no, it was a bit to see if you feed particles that are inert, that have the same buoyancy as bacteria, and you make a mixture of it, do you get an enrichment in the plant just by the recirculation rate of the sludge? So maybe the question is, all your wastewater treatment plants that you investigated in Denmark, do they all have the same sludge recirculation rate in the plant?
More or less, yes.
In the end, if it's, I'm a bit sharp in saying everything is governed by the immigration, it would mean that it's just a transport phenomenon. There is no biological phenomenon happening. It's just transport phenomena, accumulation, advection, recirculation, that would govern what you have in the plant. And maybe at a certain point, there is a need to make some real tests by feeding something in the influent that is inert, that would behave like a bacterium without growing.
Yes, we have thought also about the possibility to test with lab scales, in lab scale, the possible effect of immigration. Yeah, it's a bit tricky. It's a bit of a tricky setting. I think we just we go for full-scale plants because they are just available to us, and also the plant operators are so kind to be part of our investigations that we would rather go and see in real life what is going on and try to get the best out of the samples. But I do not exclude the possibility that we could test in lab scale also the impact of immigration.
Very shortly, because for the water phase, we can put fluorescein to track the water and to make a tracer test. Is there a particle tracer that could be used to follow the bacterium from the influent through the plant, etc.? So maybe there is something like that to imagine as an experiment.
Mm-hmm. Yes.
I've still a problem with that the immigration is so dominant. According to you, first you see that most of the organisms which are in the influent are decaying in the treatment plant. That's also what you show.
Mm-hmm.
Did you look, this is probably for a large part because a significant fraction of your influent population originally lived in our guts, and it is used to 37 degrees and not to the 15 to 20 degrees. And aerobic conditions, so a significant fraction will decay. An organism like, so that's logical. An organism like, what was the Teotrix? Microtrix is a bit a low DO organism. Could the correlation you see between the amount of microtrix in the influent and the amount of microtrix in the treatment plant also be related, that just the general climate conditions during the year induce more or less microtrix growth, either in the sewer system or catchment area up front and in the treatment plant at the same time, and it's just a correlation of local temperature changes or local temperature changes which induce local food change, local hydrolytic condition changes which induce different microtrix behavior.
Well, I hope I'll be able to answer to this question the next MIWI, because we have the data set actually to look into this question. It's definitely something we were looking into. So I cannot answer right now, but it's something we have in mind. Yeah, definitely the yearly sampling can give us also information about seasonality in activated sludge, in influent, and then I can come back to that with an answer.
It might in this context also be interesting to look to granular sludge reactors, because the granular sludge reactor has a significant population of granules and a smaller, maybe 10%, usually in operational condition of flock-like material. And the solid retention time of the flock-like material is around 4 to 5 days, and of the granular material is up to 2 or 3 weeks. But you see that the microbial, that also there, if you look to the immigration, most of the organisms which are present in the influent are net decaying inside the treatment plant, like you find. But you also see that flock-like material is, of course, you might expect that, still some similarities with the influent population, but the granular sludge population is very distant from the population from the influent, so very selective. And you see hardly any of the organisms which are in the influent popping up inside the granules as anything significant. At the same time, some organisms which are in the granules you can find also in the influent, but my response there would be everything is everywhere, so also in the influent.
Yes, yes, that's something also we have thought about, and I think it's a matter also again in the equilibrium, we can call it, between selection and immigration, right? I think these are the two main forces at play. I'm sorry, also drift, of course.
Yes, drift's important.
But of course, where in those systems where the selective pressure it's very strong, such in granular systems, then I would expect, as you have found, a lower impact of immigration. While in the activated sludge system, suspended growth, flocks, in that case, maybe the selection strength is not as strong, and that's why we see a stronger effect of immigration. That's my explanation to that, my point of view.
Yeah. No, it could be, you see also in the granular sludge reactors that the stability of the population in the flocculant fraction is much more dynamic than in the granular fraction, despite that the diversity in the granular fraction is very small compared to it. So again, a small microbial diversity leads to a higher stability than a high diversity, which leads to a higher variability also. Was also one of the other speakers coming to that conclusion. Per,
yes,
you have the last word, because I think that we go for...
Just a question actually to Ezequiel. You showed this about disturbance and very nice experiments with the lab scale reactors or the small model systems. So my question is, you showed really a strong effect of this disturbance and development in community and so on. Let's assume that Julia, she's correct, that mass immigration really means a lot. When we have a huge disturbance, then there would be a chance that you have a lot of other bacteria ready to take over the niche. So the question is it possible to translate your experiments from the big disturbance studies with this without immigration to a real world with immigration? What would be your take on that?
Thanks for the question, and it's also something I thought about. Dispersal is one of the processes that can be stochastic, right? Mostly people consider it as a stochastic, sometimes it is not, right? And then in the system that I tested, I removed that out of the question because it's normally very hard to assess. And then you guys are trying to do that, right? And the findings were mostly related to structure, but not necessarily assembly. So I would say experiments like the ones we did at Celsi give you the basis of, okay, what could be happening when you have the other stochastic assembly mechanisms operating, right? Particularly drift, priority effects, perhaps because it's from the seed bank, you do have seed bank populations, and then you could have also a little bit of speciation even in a short date span, because the mega plate study showed that you can have speciation in 11 days. So it could be, and then for that more research is needed. Something very interesting that we have related to the studies that we're doing now for microbial protein related to production, which was shown yesterday by Bethatri. Our influent is non-sterile. It has a microbial community, and that microbial community has an effect on what we see in our bioreactor, but it's not all the same by all means. And that makes sense from a point of view of if you understand the systems that you have outside and inside the reactor. That's why I think our observations and correlations in this case of immigration has to be accompanied by some understanding that could be provided by theory, right? Otherwise, it becomes just a correlation. But if you have a reason that could be theory, like in our case, intermediate disturbance hypothesis, and then a assembly structure, then you can say, okay, maybe this framework actually can work and goes beyond a correlation. That we might take.
Good.
Okay, thank you. I think we will install a break here till 4:30. And during the break, you will have to do some task, and David will announce that.
Right, simply, if you look at the chat, there is a link. You have to survey the posters and vote for your best posters with constructive alignment of the evaluations. So you have to evaluate the posters, and then there will be some poster awards tomorrow along with the award session. So have a nice break.